One of the most devastating aspects of stroke and severe head trauma is that the neurons we lose are actually never replaced. Depending on the site of injury, patients can suffer from a permanent loss of crucial motor or cognitive functions such as memory and language. But the brain does have the ability to produce new neurons following injury. It contains zones of special cells called neural stem cells that can produce neurons following injury. Unfortunately, only a small fraction of these neural stem cells activate upon injury. As a result, only few neurons are produced, and even fewer manage to survive and repopulate the damaged site. Instead, the site gets filled with cells called glial cells, literally the glue of the nervous system. So, how can we boost regeneration? The first step is to understand how injury activates neural stem cells. To do this, we turn to fly and mouse models because their brains, just like us, contain stem cells that can produce neurons upon injury. Following a deep investigation, we found that the key is a tight cooperation between neurons and glia. More specifically, we found that neurons in the injury site produce a molecule that turns stem cells on. But that molecule can't reach the stem cells on its own. This is where the glia come in. Glia in the same area secrete a transporter that carries the molecule to the nearby stem cells, where it triggers the generation of new neurons. Now that we know who the key players are and how they communicate together, we have a shot at giving neural regeneration a boost. First, we need to verify that a similar mechanism also exists in the human brain, and then we can start thinking about how to translate these findings into therapies.